Okay, so this is the fourth week of GIDAS. And so this week we're going to be going over the Genes and Health Contest, GEO2R, StringDB, and then we're going to start KEGG pathways. So Grace, do you want to talk about this really quickly?
So AmeriCorps is doing a Genes and Health Contest, and it's basically what we're learning right now and some other stuff, which we could teach you guys if you guys are interested. It's February 5th, so it's in like 2 weeks, and it's all online and it's free. So even if you don't really know the stuff, it's good to just see how like competitions are and just to see what you know. And there's gonna be like a talent show, which is, I don't know, maybe it'll be cringy, but you can see people's talents.
Yeah. And then if you guys want to join, or like if you guys want to participate, we can also give you the study material and then like register you guys. And then there's like 3 parts to test. It's like multiple choice, research, and then the third part is just like presentations and the talent show. So just a quick review of what we did last week, because I know some of you guys weren't here last week. We went over GEO2R. And so here are some like terms that are like good to know when we're talking about GEO2R. So the first one is up and down regulated genes. So an upregulated or an overexpressed gene is a gene that is more active than normal. And then a downregulated or underexpressed gene is a gene that is less active than normal. And so log FC, basically, when we talk about log FC, it means log fold change. And it indicates whether a gene is overexpressed or underexpressed in the experimental samples, the disease samples compared to the normal samples. And then the absolute value can tell you how much larger or how much less the change is. And a larger absolute value indicates a more significant result. And then p-value is just a statistical value that basically tells you if the results of your experiment were completely statistical, like by random chance, or if there's actually a correlation between the 2 variables that you're testing. And so you always want to make sure that the p-value is less than 0.05, and this guarantees that it's not just random chance. So basically, what we decided was we looked through the schizophrenia data sets from a couple of weeks ago, and we decided that it would be better to do bipolar disorder because this one just has better correlation compared to the other data sets that we could find. And so, yeah, here's just a summary of what the data set we picked is about. There's like a group for bipolar disorder and then a group for like a healthy control group. And then, yeah, there's just like a summary about what is in the data set. And then also they have like patient like descriptions for each like data set value. And then let me just pull up the data set. So here is the data set. Here's where you can analyze it actually. Let me send the link in chat. Okay, do you guys see that? So if you click on it, it'll take you to where you can analyze the data. And so here you can see that there are different accession numbers, and there are also like there's a title for each like data value. And so it tells you whether the patient that is being described here is bipolar, if they are not bipolar. So healthy control is not bipolar, and then bipolar is bipolar. And it also has extra information about like the gender, characteristics, brain pH, side of the brain, and then if they received lithium treatment. And so when you're analyzing the data here, what you want to do is you want to define the groups. So if you click define groups on the top, we can make 2 different groups. So one group we can call bipolar, and then another group we can call the control. And so basically what you can do is you can click on all of the samples that are bipolar. So this might take a little while because there are a lot of them, but you just want to click and hold, and then you can pick all of these samples that have bipolar disorder. So let me just do that really quickly. And then you can put them all under the bipolar disorder group, and you can do the same for the control group and put them under the control group. And so we're basically going to analyze, oh, wait a second. Oh no. Okay. I'm gonna have to redo that really quickly. Yeah, we're basically going to analyze both of the groups and see like more about like what I was just talking about the log fold change, and then also the correlation between the two variables. Just make sure that you're only picking the bipolar disorder groups.
Hi, Shruti.
Hi, Shruti.
Shruti!
Hi.
Okay, so now I'm just gonna pick the control groups really quickly. Okay, I accidentally picked
Oh wait.
Yeah, I
Did you like sort it first?
Oh, like based on
Yeah.
bipolar control. That's smart. I should probably do that. Okay, it's fine. Let me just. Okay, there. Okay, so now once you've put like all the samples into like their respective group, you can go to the bottom and click Analyze. And so here it'll analyze the data, and it'll come up with a couple of graphs on the data. And then over there, what you want to do is you want to download the full table. We don't have to do that because I think, Grace, you've downloaded the table already. Yeah. So she's downloaded the table already, and she's also actually like sorted the data and put it into a spreadsheet. So here's the spreadsheet. Okay, yeah, so also here are the graphs. And so here is a like a plot of like the log fold change. And so basically there's like you can also over here see like the bipolar disorder and then the control groups on like a UMAP plot. So there are a bunch of different graphs that we can use here to like look at the relationship between the variables. But you can also download the table of all of the data. And this is the original table, but then what Grace did was she looked at which p-values were less than 0.05, and she kept those. And then she also looked at which, like in the gene bank accession column, which ones had NM at the front, and she selected those genes. And so now what we do is we take all of the genes that like Grace sorted out, and we're going to put them into string DB, and we're going to use that to look at all of the genes in a map. So is everybody okay with this so far? Like no questions? Okay, let me just put the link for the downloaded table in chat. So here's the link, and then just go to the second, like step 3 on the bottom, and select all of the genes that are here. And so you only want to select the first 250. So, but I think, wait, Grace, have you looked at like this, the blank spaces?
You can just select the whole column is like 330 something. You can
Okay.
just select the whole column. It's okay. That's it.
So after you select the whole column, then, okay. You can copy all of these values into string DB. So let me put the link for string DB in chat really quickly. So after you've copied all the values in that column, go to the link for string DB. So it should take you here. And then you can go to search and then do multiple proteins. And you want to just like paste all of the, oops. You want to paste everything that you copied from over here into string DB. So it should look like this. And then for organism, put Homo sapien, because in this data set, everybody that was a part of the experiment was a Homo sapien. And so it's right here. So is everybody good with this so far? Okay, we'll just do search then. And then when you get to this page, you click continue. And so click continue again. And then it will give you a map of all of the genes over here. And it should look something like this. And then when we go here, so what you want to do is you want to check off evidence. So if you go on the very bottom of the page and you go to settings, you want to, instead of confidence, you want to click evidence, and this will change the lines to colored lines. And that also indicates like the type of interaction between the genes. And then you want to hide disconnected nodes in the network, because we don't want to look at genes that have no correlation to any other genes. And then let me click update over here. And that should make the map like clearer. And then we can see like these genes that have a lot of connections are probably genes that we want to look at. So is everybody good to this part? Like nobody has any questions? Okay. So if we look at the analysis section here, we can see a bunch of different biological processes. And then we can also see KEGG pathways. And so what we want to do is, for example, if you're looking at biological processes, there are like diseases and things here that are also been listed. And so if you click on, for example, okay, let me just click on a random, let me just click on cellular process, for example. All of the red genes are like also a part of cellular process. And so that's how you know what genes are a part of like multiple biological processes, and we can take a look at that too. But something we're going to do this week is look at KEGG pathways. And so here are a couple of pathways that we're going to take a look at. So yeah, Grace, do you want to go ahead and start talking
I'll do
about
this.
do you want to share your screen, or should I just?
Can I share?
Yeah, sure. Is what KEGG pathways are. So basically what we saw over here, what we saw over here was, yeah, so what we saw over here with KEGG pathways, for example, if I click like Parkinson's disease, what it's going to show me here are all of the genes that are related to Parkinson's disease are in the same like pathway as Parkinson's disease, and are also in the pathway for bipolar disorder. And so this is something that we can take a look at through KEGG. Basically, KEGG is a database of resources for understanding like biological systems like the cell. And then what we're looking at is specifically like molecular data sets and genome sequencing. And so that's what we're going to use it for. So I think Grace started here. Can you share now?
I cannot share.
Okay, do you want to just talk about it? I can like move my
Oh,
screen.
you haven't allowed me to share.
Oh, okay. Wait, let me make you host then. Cool. Yeah.
You see? Okay, so here is my string DB map. And if you go into analysis, did you already talk about this, the KEGG pathways? So we're gonna be looking at the proteasome because why not? Um, so if you like click on the specific genes, you'll see like their information, and then you can read a little about it. Why is it not working? And if you read into it, it's like blah 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 blah, some useless stuff, and then it's focused on the degradation of ubiquitinated proteins. So since like I did some research into this, and then these, where'd it go? Ubiquitin proteins, and like this pathway has a significant, what am I saying? It is connected to the ubiquitin protein. So then if you click on like this HSA number, it'll actually take you to KEGG pathways, and then you'll see like the proteasome pathway in like how it works. So here you could see the ubiquitin mediated I'm not going to pronounce that. And you can see how like the gene works, because it gives you like multiple genes and how they're in the process. And as well as that, you can also go to KEGG pathways. And okay, if you just search up like neurodegeneration and click go, and you go to the map and click the map here, it'll show you like the pathways. Oh, and it'll show you the pathway of like neurodegeneration, and like everything in here is sort of complicated. But then you'll see in this side there's like calcium signaling, and what we were looking at is a ubiquitin one. So if you just check it, it'll like highlight all of it for you and the pathways for all of that, and then you can see what's happening, and you can do more research into that. And also, if you're like really focusing on the ubiquitin and the bipolar disorder, you could just search up like ubiquitin and bipolar, which was in the slides. Like there was an article. Let's see. Ubiquitin. Oh, I already searched it before. Okay, there's a research paper that basically about the ubiquitin proteasome pathway in bipolar disorder and like schizophrenia. So you could use like these sources to come up with your own research. And yeah.
So this is an option of something we could do research on. And then if you look back at the KEGG pathways in StringDB, there are also like many other, I guess, pathways we can take a look at more. So we can decide that, like, Grace, do you want to decide that this week or should we decide it next week? Is it?
We can decide this week.
Yeah, I think we have enough time. So do you want to go back to StringDB really quickly?
Oh. Okay.
So is there one that you guys want to do? Because and can you like expand it really? Expand the KEGG, the arrow.
The arrow?
The arrow next to KEGG pathways on the left.
Huh?
Over at pathway. See how it says pathway in description? Yeah.
Oh my goodness. Wait, what?
No, the arrow on top of that. See the arrow?
Oh. Okay. Why does it keep doing
that? Okay.
How do you make it like? Oh, Grace, what are you doing? Okay.
So I think also strength is important. So it looks like the one that we just did has the greatest strength. Yeah, so is there one that you guys want to pick? for what you want to do research
I
on.
just learned about oxidative phosphorylation. Oh my goodness.
Yeah.
I like remembered something from bio. I'm so proud of myself.
Yeah.
Like,
So we could do oxidative phosphorylation. We could also do like any kind of disease. There's Alzheimer's, Parkinson's. We could do the one that Grace just picked, or we could do like metabolism or the other one that's on there. So is there one? Do you guys, can everybody just put in chat like the one they want to work on or like do research on?
I think it'll be easier to do proteasome because um the process was that it breaks down proteins, and since we're doing a neurodegenerative disorder, you could connect that to the proteins. So I think that would be
I'm
easy.
fine with doing that one.
You're okay with doing that one? Okay. Yeah, so is everybody good with doing the proteasome one? Just like React.
Yeah, sure.
Okay. Okay, cool. So I think we can just stick to that one. And then I can maybe put up, let me make a Google Doc really quickly with this on it. Oh, it's not letting me minimize. Grace, do you want to make a Google Doc really quickly? Or let me just make one. Give me a second.
What? Are you gonna make one?
Yeah, I'll just make one, but I'm just gonna, yeah, let me just make one really quickly. That's it, right, for KEG, for everything you wanted to talk about today?
Okay.
Um,
Is
so basically,
for the next few weeks, are we gonna do the research? Because if people are interested in the contest, we could also teach a little bit of that stuff.
yeah, do you wanna, okay, so that's up to them. If you guys wanna do the contest, we could do the contest. We could continue doing research, and then we could also teach R, like the programming language.
Yeah.
So we have like a bunch of different things that we can do. Yeah, so it's up to you guys. I'm gonna make a poll really quickly.
Oh wow. You can do polls on here.
I think so. Oh, I think I need to enable meeting polls for that.
Is this recording?
Yeah, that is.
Oh.
And I'll just cut it out. Okay, maybe you know what? Just put in chat. Do you want to do, okay, so one is learning about the R programming language. And then two is doing research, like using KEG. And then three is doing the genes and health contest. Can you guys put in chat like the number? And based on that, we can decide what we want to do in the coming weeks. And then, yeah, I think, Grace, do you want to go back to the slides really quickly?
Oh. I didn't open this.
Okay. I think I have a team bonding activity at the very end. Depends on what you guys want to do, if you guys want to do it or not. I think we have,
How the heck do I? Where's the bar thing? Oh.
okay, this meeting is going to end soon. Okay, I think everybody, did everybody put their number in chat?
I have a question.
Yeah.
What's R programming?
So basically R is like a programming language, but it's like not really like, and it's not like when you think of a programming language, it's not like that. It's more of like data set analysis and stuff. And you use like different commands to like analyze data sets, make box plots and stuff to
Oh, okay.
correlation between data. It'll help us do our research. So yeah, it's a good thing to do too.
Okay, cool.
We'll like get to them eventually.
Yeah.
Yeah, because they all support what we're doing.
Yeah.
Okay, cool.
So I think everybody wants to do 2 or 3. So, and then Shreya wants to do 2. So I think the next couple of weeks we can focus on continuing research, and then the genes and health contest thing we can do both of them, because the genes and health contest is also pretty like important. Like we'll learn a lot of actual material from that might be helpful. So okay, so that's what we'll do in the coming weeks.